Here's extension work. Figure out this.
Okay, I'm going to take a different type of approach to these talks than what other folks have been doing. Rather than present kind of a story of one specific aspect of my research, I thought I would kind of give an overview of the kinds of things that we do in my lab in the context of ecosystem ecology, so that you can get a flavor of the kinds of things that we do and why it's interesting and important and how it might relate to this project. So I think I'm the first ecologist to talk, right? So I am in a biology department, and the way that I approach the work that I do is by thinking about organisms as part of a physical, chemical, and biological system that functions to cycle materials and have energy flow through it. And basically what that means is that, say, something like a stream or a river processes materials like nutrients, and the organisms that live there have energy flow through them. So from the plants gathering energy from the sun to produce biomass that gets eaten by some other organism and then another organism. And in the context of other fields of science that are represented in our group, here's ecosystem ecology, and we're kind of situated in the middle between context at a broader scale and mechanisms at a lower scale. And first, some of the kinds of things that we do. First, we make observations, and those observations can range from very simple observations like measuring temperature to measuring the rate of something. Here we're doing a little bit of groundwater sampling to measure denitrification rate. That's an observation of a process. We also do experiments. This you can't really see, but we do things to the ecosystem to change its properties. And this is just showing what happens if you exclude carp from a stream. You get an increase in biomass of macrophytes. We also use models, and this is a pretty complicated looking model, maybe even more complicated looking than Beth's. But we use different types of models. This one is just really a heuristic model or a conceptual model of how we thought that streams and lakes would interact in nitrogen cycling. And the details of all these crazy arrows and things aren't really important for illustrating that. We just think this is how it works. And then we might go out and make some measurements of these different things to see whether or not our model is correct. I'm not smart enough to write an analytical model that describes all of these fluxes, but I have colleagues that do that. And so they would write a numeric model that gets fed data and then predicts observations. We also use statistical models to say if we want to test the difference between the inflow versus the outflow, we could use some statistical modeling to tell whether or not there's a difference. Most of the work in my lab focuses on processes that are involved in nutrient cycling in streams and rivers, and these processes affect water quality. And I'm going to give you a little bit of background on the increase in nutrients that affect water quality. This is from a picture from China right before the Olympics. So nutrients are required for all living things. Reactive nutrients or nutrients that biota can use are increasing at an alarming rate. Cycling of nutrients is complicated, and impairment of water bodies can occur at very low levels, and most nutrients are not considered toxic. Sources of nutrients and therefore management options extend beyond watershed boundaries. But I think that the kinds of work that we do would be a good opportunity to foster interdisciplinary collaboration, because really what we do is measure attributes of stream health. So I've said a little bit about nutrients. What are they? In the context of my lab, mostly we are measuring things like carbon, nitrogen, and phosphorus.
So what we do to organisms is we take them to the lab, drive them up, and measure what's in them. Carbon is found in everything. So almost half of the dry weight of an organism. Nitrogen is mostly found in proteins, and it's about 1 to 5% of dry weight. Phosphorus is important. It's found in the cell membrane that surrounds cells, and it's in nucleic acid, so in DNA. It's less than 1% usually of dry weight. And measuring these things in a ratio can tell you something about the physiological status of organisms in the system and what they might need in order to grow and reproduce. I'm going to focus now mostly on nitrogen and phosphorus because that's mostly what we study. On a global scale, most of the nutrients in the world are not biologically available. So this is a table that lists the major pools of nutrients. Most of the nitrogen is tied up in the atmosphere or in rocks. Phosphorus, most of it's tied up in rocks and soil, and it's not biologically available. How does a nutrient become biologically available? Well, for nitrogen, there's 3 processes that can take nitrogen out of those atmospheric and soil pools and make them biologically available. Nitrogen fixation is a process that bacteria can do where they take nitrogen gas and turn it into ammonium that then becomes part of their amino acids and proteins. Lightning also converts atmospheric nitrogen into an biologically available form. And then decomposition of organic matter releases available nitrogen back to the biota. These 3 that are highlighted in blue are done by humans. So by combusting fossil fuels, by producing industrial fertilizers through the Haber-Bosch process, and by changing landscapes to cover with legume crops like soy, increases the amount of biologically available nitrogen. Phosphorus can only become biologically available by chemical weathering and decomposition naturally, but mining activities can produce available phosphorus. This is a graph from the Millennium Ecosystem Assessment that shows the amount of nitrogen that is biologically available produced per year as a function of time since 1900. And highlighted here is the range of natural nitrogen fixation by bacteria. So it's around 150 teragrams per year. What these different lines show are different anthropogenic activities that occur: fossil fuel combustion, conversion to legume crops, and fertilizer production and other industrial uses have made it so that the total amount of nitrogen produced by humans is equal to that would occur naturally. So we've doubled the amount of reactive nitrogen that is cycling globally. We've done the same kind of thing for phosphorus, which is shown here. This is phosphate mining production per year. And our activities have quadrupled the amount of reactive phosphorus. So this leads to some locally observable impairments to streams and rivers. This is from the Environmental Protection Agency's Weightable Streams Assessment that shows basically the relative extent of impairment due to various anthropogenic activities. And you can see that nitrogen and phosphorus impairment are the most important ones up there. And in the report, they estimate that 1/3 of the stream miles, or 200,000 miles of wadable streams in the United States, have too much nitrogen or phosphorus. So this leads to impacts on designated uses of our streams and rivers. Here are nutrients Coming into systems, they can affect the growth of plants and algae and microbes. And these different groups of organisms, the microbes and the algae, impact these attributes of streams that we might measure, and that might be regulated by these various uses. So the nutrient and biota together kind of describe something that we call trophic state. This is a good picture that illustrates where the term trophic comes from. So a heterotroph, this guy, he is an other feeder. Means he must eat food in order to acquire energy. This stuff that he's wearing is algae. Algae are autotrophs. They're self-feeders. They use sunlight to fix atmospheric carbon dioxide into organic matter that they use for energy. You might be more familiar with these terms, oligotrophic, or a low nutrient system such as Bear Lake, compared to a eutrophic or high nutrient system. This is Lake Taiyu in China, and you can't really see this is an algal bloom. So in streams, trophic states are usually characterized by the amount of chlorophyll, but the amount of chlorophyll that you see in the stream depends not only on the biomass, but also the rate at which those organisms are cycling materials and acquiring energy. And we usually don't consider the heterotrophs, the guys that don't produce their own organic matter. But both of these states, autotrophic and heterotrophic states, are affected by things like temperature and nutrients. So here, for example, nutrients affect the rate of primary production by algae. They also affect the biomass of algae that you find. Nutrients affect the biomass and rate of processing by bacteria, heterotrophs. And in my lab, we measure all these different kinds of things that are in the boxes. And at the ecosystem scale, or at a reach scale of stream, we measure things like how much primary production there is and how much respiration there is. So I'm just going to give you a few snippets of the kinds of things that we do. And it's mostly summed up in this cartoon, where Calvin and Hobbes see a little trickle of dirt and they say, "Woo, our afternoon just got booked." So the first parameter that we measure is called ecosystem metabolism. I alluded to that in talking about primary production rate and respiration rate. It's a measure of how much energy is being processed at a reach scale. And we do that by basically making an oxygen budget for the reach and deriving respiration and primary production rates from that. And what it reflects here would be the algae. What do the algae do to the oxygen, do you think? They produce it. So what algae are doing is they're taking carbon dioxide and water and making oxygen and organic matter. The heterotrophs are basically doing the opposite. They're burning organic matter with oxygen in order to get that energy. So together, those two things are affecting the oxygen budget, and we can estimate that from the oxygen record. And this is some data that Eric Duffin gave me, so it's nice to see you here. But it's from 2 sondes that were deployed in the Jordan River 5 or 6 years ago, just showing what the data look like, basically. And this particular site, the oxygen goes from around 70% saturation to above 140% saturation, which means there's a lot of oxygen being produced. And that particular reach, if you do the calculations, has a greater rate of primary production than this site, which is farther downstream and is much lower in saturation and rarely exceeds 100%. Another thing that we've just started measuring is biochemical oxygen demand. And it's typically measured by sticking some water in a bottle for 5 days and then measuring what happens to the oxygen. So it's basically like measuring a respiration rate. So how much oxygen is lost over that time. You can also do some different kinds of manipulations to get at different types of BOD, but for right now, I'm not going to talk about that. I have an REU student who was doing some nutrient bioassays in the context of BOD, where he's basically adding fertilizers to BOD samples to see how that affects oxygen consumption. And he was doing this for a different project, and he was using sites around Cache Valley as development for protocols. So we don't have a whole lot of data, but he added either nitrate alone, ammonium alone, these are different kinds of nitrogen, phosphate, or nitrogen and phosphate. And in some systems, like in the Little Bear River, this was up high by, what's that town called? Yeah, not quite as high as Avon. Paradise, yeah, by Paradise. That the amount of BOD in the water doubled in response to these different treatments, but there wasn't a difference between the treatments. In contrast, Bear Lake did not really respond significantly to the nitrogen treatments, but it increased by threefold in the phosphorus treatments. And the point here is that nutrients can stimulate the amount of oxygen that organisms consume. This is demonstrating that the heterotrophs in the system also can respond to added nutrients. We do the same kind of thing for the primary producers using things called nutrient diffusing substrates. That's what NDS stands for. And we do the same kind of thing, adding nitrogen or phosphorus or both. This is what they look like. They're filled canisters filled with agar, which is kind of like Jell-O, that have the different nutrient treatments. And then you measure how much chlorophyll is produced. And if the algae are responding to nutrients, they should increase the amount of chlorophyll they have. And this is just showing some data from East Canyon Creek above and below wastewater treatment plant, showing that they're responding to nitrogen and nitrogen plus phosphorus, but not to phosphorus. And there's no influence of the wastewater treatment plant. The more complicated types of measurements that we do are in the context of something called nutrient spiraling. And these involve tracer tests like Beth was talking about. And nutrient spiraling describes the coupled transport and removal processes of nutrients. And if you think about a nutrient cycle, you have something like nitrate that gets removed by algae. And then when that algae dies and decomposes, it completes a nutrient cycle. But because we're talking about streams or rivers, these nutrient cycles are affected by transport. So the cycles look more like a slinky that we call a spiral. And this parameter, S sub W, is the uptake length. It describes how far an atom of nutrient will move on average before it is removed by the biota. And this parameter here, V sub F, is the mass transfer coefficient that describes basically how fast those organisms are removing nitrogen. So we do tracer tests that have a conservative tracer like bromide or, in this case, rhodamine dye, and then some fertilizer, and we measure the removal of those nutrients relative to the conservative tracer. We also are starting to evaluate these tracer tests in the context of an enzyme model called Michaelis-Menten dynamics, or Michaelis-Menten kinetics, but I'm not really going to talk too much about that. But I think that what these kinds of measurements can tell us are how important is a particular nutrient for this system that we're considering? How fast do they remove that nutrient? It's a measure of the stream's ability to self-purify. And I think we can use it to tell, for a given stream at a given time, what's the maximum assimilative capacity of that particular place. With the idea being that if we exceed a particular concentration for a long period of time, then biological changes will start to happen that could involve lots of periphyte growth or something like that. So we've done this kind of stuff. We started out doing this kind of stuff on little dinky streams, but now we're doing it in rivers. This is the Green River. It's running at 24,000 liters per second. I don't know what that is in CFS. 500? And this is just showing what the data look like. And the Green River is green on the bottom, and it's got really high demand for nitrogen. So relative to other streams, this value is high. And in fact, we weren't even able to detect what we added at our lowest monitoring place. So these are slugs of stuff that we're adding. And what happens as that material passes through is on the leading end and on the tail, the concentration that you're measuring is low. And it peaks out at some concentration. And biological responses are affected by the concentration. There's only a certain number of enzymes that are in the stream that can process that nutrient. And so we can use these data to explore the concentration dependency of these parameters. And I'm not going to really describe this, but just note here for these open circles, this is describing how fast organisms are acquiring the nutrients. Here's the peak of the injection, really low rates of uptake. Here's the tail where that uptake is much higher, much faster. So if we express these in terms of a rate, these follow what enzymologists call Michaelis-Menten kinetics. And we can use them to predict the concentration at which the biota becomes saturated and unable to acquire more nitrogen or phosphorus. So I think that's an interesting potential measure of the health of a system. So just to sum up, we use a whole bunch of different kinds of tools, and they describe aspects of how the stream functions, and some folks call these attributes of ecosystem health. And I just want to thank the collaborators that have worked on this project. I forgot to add Matt because I used an old slide. But I'd take any questions if you have any.